Hello, everyone. Welcome to today's webinar brought to you by Microbial Insights. This will be on harnessing synthetic biology on Mars, and then challenges and opportunities that arise with it. We appreciate that you're taking the time to join us today. It is my pleasure to introduce our esteemed speaker, Dr. Tiffany Vora. Dr. Tiffany Vora is an entrepreneur, a scientist, a writer, and thought leader who lives in Silicon Valley. She earned her undergraduate degrees in biology and chemistry at New York University and her PhD in molecular biology at Princeton University. Among other experiences, she was formerly a professor at the American University in Cairo, an instructor in bioengineering at Stanford University, and the faculty director at Singularity University. Tiffany is currently the Singularity Fellow in Biotechnology at Singularity University, a non-resident senior fellow of the Geotech Center of the Atlantic Council, a regulator contributor to EY's executive learning programs, and the VP of Innovation Partnerships at Explore Mars. Now, before I let Dr. Vora take it away, I would like to remind you all, as always, this webinar is being recorded. It will be available in the resource section on Microbial Insights website by the end of today, if not this week. So don't worry, we'll also be sending out an email with a link of a certificate of attendance following this webinar to those that have registered for it. After she is done, there'll be some time for questions, so feel free to type your questions as the presentation goes on, and I'll try to read off as many as I can before that time comes, so that way she can get you your answers. So join me in welcoming a true advocate for sustainable solutions, Dr. Tiffany Vora.
Thank you so much. Good morning, everyone. I'm absolutely delighted to be joining you. It's morning in California. And today we're going to be talking about one of my very favorite things, which is the potential for harnessing synthetic biology to enable humans to thrive as they live off Earth. So when I talk about synthetic biology, I'm taking the perspective on it that SynBio is merging biology, engineering, and other disciplines to design and construct new biological parts, devices, and systems that, to our knowledge, don't exist in the natural world. And I'm also mean SynBio to include redesigning existing biological systems to perform specific tasks. And it's that combination of capabilities and possibilities that I think makes SynBio so appealing as a potential for supporting sustainable human exploration and settlement of space, specifically Mars. So I'm going to focus on Mars today in my role of VP of Innovation Partnerships at Explore Mars. I obviously have a Mars bias, but I think that a lot of the things we're going to talk about today really work for low resource settings in general. The details will change, but whether we're talking about Mars, whether we're talking about a space station, whether we're talking about the moon, whether we're talking about somewhere farther away, or on low resource settings on Earth, I think there's just a lot of value had here. Of course, there are also challenges. So I think with this audience in particular, I don't have to sell you on the value of synthetic biology, why this is so cool and why microbes are absolutely the best. I did want to show you, however, the way that I tell this story to folks who are not microbial scientists, who are not in the biotech field. What I usually ask them to do is I ask them to imagine a manufacturing technology that is self-replicating and self-repairing. It's modular. It's very flexible in terms of its input. It's programmable and reprogrammable with great precision. It's remarkably resilient. And this manufacturing technology can be stable for years to centuries to millennia if you keep it dry and cold. And then, of course, the big reveal is this technology is life. So at Explore Mars, we are a nonprofit based in the United States that is building a large stakeholder community to advance the human exploration of Mars and beyond, no later than the 2030s, with the idea of building a sustainable and inclusive future for as many people as possible. And in my role, I'm particularly interested in technologies that maybe today people don't think of as being space technologies, but I believe in 20 years, 30 years, 50 years, whatever far horizon we're looking at, that these technologies and these innovation spaces really have the potential to transform what human life off Earth looks like. And really, I spend a lot of my time answering the question, why should we spend money on space when we have so many problems here on Earth? And my answer to that is that the problems that we have in space are the same problems we have on Earth, maybe in a more extreme form. But whether you're talking about renewable energy, whether you're talking about closed loop ecosystems, whether you're talking about education, making more humans, keeping humans healthy, having enough food, having nutritious enough food, these are all the same problems between Mars and Earth, which is why I'm so excited to look for areas of overlap. And so as a result, at Explore Mars, we're particularly excited about these innovations that can benefit people both on and off Earth. And that's why for the last couple of years through Explore Mars, I spearheaded a working group that focused on the promise of synthetic biology for the space industry. So we had deep subject matter experts in our group. The folks who ended up joining the core of the working group were Dr. Lynn Rothschild from NASA Ames and Brown University, Dr. Erica De Benedictis, who is the founder of Pioneer Labs and was also has been affiliated with the Crick Institute, and also Dr. Niels Averish, who when we started this project was at Stanford and is now at the University of Florida, and he's a major contributor to the CUBES project. And among other questions, we on this working group asked, what are the bottlenecks today to progress in synthetic biology for space? Which of course, we want to also know, how do those bottlenecks enable synthetic biology on Earth? So we published a paper on this in 2024 in New Space, and I'm going to tell you a bit about what we looked at, where we landed, and where we think there's a lot of possibility for SynBio in space. Now, I'm a molecular biologist by training, and my innovation work is focused on the biotechnologies and all the sectors that it enables. I imagine that you all share my bias that biotech is absolutely the best industry and the best field to be working in right now. And I think one of the major reasons that we're at this exciting moment is that the barriers between the living and the non-living world are breaking down because of exponential technologies in computation, in sensors, in biotech itself, in chemistry, robotics, automation. All of these things are coming together to bring us to this moment where increasingly biology is technology. Now, to us, that's not a surprise, but again, to folks who are outside of our field, I go around the world speaking to people about tech and about biotech, and the vast majority of them will say something to me like, my business has nothing to do with biology. Why should I care about this? And always my response is, do you have a body? Do you have people you care about who have a body? Do you eat food? If that's true, then understanding the revolution that's happening in biotechnology today is very important for understanding the future, not just for yourself as an individual, but as a family member, as a member of a community, as a member of an organization, as a member of a country, and then of course, at the species level, as a member of the human species. So for me, what's really exciting is that today is the most boring biotech is ever going to be. Our tools are only going to get faster, cheaper, more powerful, and more accessible as time goes on, which opens up a whole series of innovation spaces for people like us to play in. A beautiful example of that, of course, is biomanufacturing, biotechnology, and the bioeconomy. As I mentioned, I'm American, and in late 2022, President Biden's administration issued this executive order basically calling for a whole of government approach to advancing biotech and biomanufacturing across innovation spaces, health, climate, energy, food, agriculture, supply chains, and of course, national security. And the US has not been the only country to do this, not by far. It's a huge business opportunity in the synthetic biology and biomanufacturing spaces around the world. So in March 2024, the EU published their strategy, which they called Building the Future with Nature. And so basically, the USA, Germany, China, the UK, and other nations have published dedicated roadmaps for their bioeconomies. And many other countries around the world have initiatives to grow their own country's bioeconomy. So the map that I'm showing you here on the left is from December 2023. It's already out of date, of course. We know, for example, that there are major players in the Middle East. UAE is active in biotech. Saudi Arabia has a plan to be a biotech leader in their Vision 2030 plan. And I'm really excited about this because these bioeconomy strategies signal an intent, and hopefully an intent to fund innovative products and processes that can impact our existing processes around the world. I believe that biomanufacturing and the bioeconomy can change the availability of input raw materials to overcome scarcity or to switch over entirely, for example, from petrochemical stocks. Remember that point, we're going to come back to it. And the big promise here is to improve sustainability and resilience of our businesses and our products and our services around the world. Now, a big goal for the bioeconomy, as I said, is to improve resilience and sustainability. And I like to think of that across the UN SDGs. So we're talking about protecting the environment, we're talking about promoting socioeconomic development, and also supporting industry. And that maps to many of the UN SDGs in ways that benefit businesses and workers and individuals and the environment. And so I believe that biotech is really important towards making progress toward achieving the UN SDGs. Now, I promised that we were going to talk about space today. And so here's the link. Everything that I think is true about the circular bioeconomy on Earth is also true for a bioeconomy in space over both the short and the long terms. So let's go now and talk more specifically about what this could look like for space and where the challenges and the opportunities are for synthetic biology in particular in space or off Earth, let's put it that way. So in the off-Earth human life community, we've for a long time had the concept of bioregenerative life support systems, which are BLSS. And they're also known as closed or controlled ecological life support systems. So these types of systems have been explored since the beginning of the human space exploration era in the 1960s. So one of these bioregenerative life support systems is made up of several interconnected compartments that are based on organisms whose wastes represent the vital inputs for the other compartments. So you've got biological producers, consumers, and recyclers in this type of system. You could have animals or insects also added to these loops, not just plants and microbes. These could be protein sources or, again, manufactories for making the components that you need. And also, it was realized early on that there are strong psychological benefits to having non-human living things in isolated environments. Plants are an excellent example of this. And so there have been several large-scale ground-based demonstrations that have tested these closed-loop systems with humans in the loop. There were some in Russia, famously. There was one in the Biosphere 2 in Arizona in the USA, where I get to go next week as part of the Analog Astronaut Conference, so that was exciting. There was another closed ecology experiment facility in Japan, and also China had one called Lunar Palace 1. And then there have been other places on Earth that have tested independent functions, like gray water recycling at the Concordia station in Antarctica, and also food production was tested in the Eden mobile test facility on the International Space Station. So there have also been lots of analog missions that have looked at these components in Russia, through NASA, through NASA's various things, and then of course, through the High Seas simulator in Hawaii, and then the Mars Desert Research Station in Utah, which is a Mars analog place where I actually did 2 Mars missions there. I've also participated in an analog simulation up near the North Pole in the high Canadian Arctic. So this work has been going on for decades. It's really very interesting and very promising. You might have also heard of a BLSS from the European Space Industry Agency called MELISA, which is a microecological life support system alternative. That started in the late 80s. And this one is a connected system of 5 components, where on the ISS, they've used this system to cultivate spirulina, a cyanobacterium, in a photobioreactor that was able to convert carbon dioxide into oxygen and biomass. And there's ongoing ground-based testing of this going on still in Spain. So lots of really interesting work going on here. So if we go back to the concept of these bioregenerative life support systems, I want us to focus the rest of our time here, thinking about microbes. So as you can see from this really great map, the microbial systems in these bioregenerative life support systems, that's just one part of the role that microbes can play in our future in space. So microbial studies in space and other extreme environments on Earth have shown that bacteria and fungi can adapt to off-Earth conditions. Now, some of these changes, like biofilm formation, can negatively impact astronaut health and spacecraft integrity, so that's in a negative way. But others can promote self-sufficiency and sustainability in space, particularly with human intervention. So I think that we should be including microbes in the planning, decision-making, and mission design for long-duration human space missions, not just astronaut microbiome studies to protect against infections and immune system dysfunction and bone deterioration, which, to be clear, I absolutely think we should be doing. But I think we should also be looking more aggressively at biological in situ resource utilization to incorporate microbes to act as radiation shields, to create electricity and fuel, and to help us establish robust plant habitats for fresh food and for waste recycling. So most important for today, we're going to focus here on the idea of bioengineered microbes, how we can use microbes to produce the things that astronauts will need for long duration or maybe even permanent missions. So that's the far off vision. And to really zoom in closer to take a look at the potential benefits of SynBio for the exploration phase, in particular, of human activity on Mars, our Explore Mars working groups took a look at the 17 NASA technology taxonomy areas from 2020. And we think that SynBio has strong potential for 3 of these areas in particular. I'm showing you here. So exploration destination systems, human health life support and habitation systems, and materials, structures, mechanical systems, and manufacturing. Those are the big taxonomy areas that we think SynBio is particularly potentially well suited for. Now, of course, there are non-bio-based solutions at work in these technology areas, but bio-based solutions have advantages that non-bio solutions lack. So I started out this talk by pointing out that biology is self-perpetuating and modular, flexible, programmable, resilient, and surprisingly stable. I think there are 2 more affordances of biological systems that are particularly important for space. The first is that life has the power to help many of the upmass constraints of space travel. So we just can't afford to take all of the stuff we need with us. It's too expensive to launch all of that mass into orbit and then beyond, for example, say to Mars. So with synthetic biology, we could be using or reusing resources and basic feedstocks that are available to us en route, right? This is loop closure, things like waste or packaging. And SynBio also empowers us to use resources that we might have at our destination. So that's known as ISRU, or in situ resource utilization. That's stuff like water, which we increasingly understand is available to us on Mars or on the poles and the moon, atmospheric carbon and nitrogen, and minerals on Mars in particular. Which means that instead of trying to store your food and your medicine for more than 2 years, which is how long a round trip mission to Mars will take, you can make what you need when you need it. And instead of storing all the things, you're able to have fresh versions of them. So I just want to come back for a second to that 2 year time point, because I think that's really important for us to remember. Mars is really, really far away. And it sounds completely ridiculous when I say it out loud but for orbital mechanics reasons, you have to leave during certain launch windows when the distance between the two planets is at the closest, because you want to minimize the amount of times that humans are exposed to the deep space environment. Space is not good for humans. It's not good for biological systems in particular. So the shorter the journey, the better it is. And once you land there, you're not just going to land and leave, you're going to land and stay for a little while, because again, you want to wait for the planets to come back to that position that's as short as possible for the journey home. So again, we're talking about 2 year missions, 2+ years, which is a really long time to keep your food nutritious, to keep it stable, to keep your medicine stable, and to keep human health in good shape. So thinking about using biology as an important platform for this going from a take to a make paradigm, you could use biological sources of materials for off-Earth construction. So for example, you could look at mycelium-based biocomposites, right? Mycelia being the root-like structures of a fungus that have these masses of branching filaments. Dr. Lynn Rothschild from our working group, she's investigated using mycelium biocomposites for building in extreme environments on Earth and also in off-Earth environments. So to direct and support filament growth, her group 3D prints a scaffold that they can coat with nutrients and agar. And then what they get are these really beautiful and complex 3D structures that get built. Folks are also working on using Martian regolith. Dirt is the wrong word for what they have. Soil's the wrong word for what they have on Mars, because it's not made by plants, but the rocky substrate is known as regolith. You could potentially also use Martian regolith as the main structural mass of a building for living on Mars. So here I'm showing you mycelium that's been grown on a Martian regolith simulant, not real regolith, but simulated regolith, with only a minimal addition of in situ produced biomass or nutrients that act as a binder. So what you get is some pretty stable materials that enable you to construct inhabitable surface structures with various material properties and functions. So, and there's many, many, many more examples. We'll come back to a couple of them in a few minutes. But again, the idea here is to take advantage of biology and synthetic biology, thinking of DNA as both a coding language and a parts list that is now essentially open source to us, thanks to DNA sequencing technologies and DNA synthesis technologies, which of course enables us to either move genetic modules around from organism to organism, or to use gene editing technologies like CRISPR to go in and change individual, say, cis-regulatory elements or other types of protein coding areas of the genome to be able to change the biology of a living cell to do what we want it to do. And that could even include putting in functions that, to our knowledge, have not been used by nature today. I'm going to come back to that. So the mental model that I usually ask my non-biologist listeners to use is to think of the living thing as the hardware and the DNA code as the software. And so the modules get put into the hardware, and then it basically compiles the DNA code and executes it, and you get a structure or a function or a set of molecules that you can use for something. I tend to think about synthetic biology in three major buckets that we can use to make the outputs that we want. I mean, again, this is the beauty of biology as a flexible technology. And we can think of it again, especially for our non-biologist colleagues, by thinking of the cell as a living factory that's filled with machines like DNA and proteins and other molecules that make the thing that we want. So we can do this in big tanks with live cells, which is precision fermentation. I tend to think of precision fermentation for medicine, materials, and nutrients in particular. And this is a really beautifully established field here on Earth that I'm expecting to see a lot more action in the coming years. We can also think about cell-free synthetic biology, which is breaking open the living cells and just keeping the biological components of the cells that do the biological manufacturing. It's usually proteins and some other molecules. And then sometimes the living cell itself or a group of living cells is what we're after. And so when I think about whole cell synthetic biology, I'm thinking about probiotics or cell therapies. I'm thinking about living sensors. I'm thinking about lab grown meat or seafood. And so the key is that in each case, the DNA software, of course, has been manipulated by people, and that's what makes it synthetic. When I'm talking to non-biology audiences, I always make a big point of saying that synthetic does not mean good or bad. Synthetic just means that humans have worked on it. I also usually point out that just because something is natural doesn't mean it's good for you, right? Arsenic is natural, doesn't make it good for you. So when we're talking to the public about this kind of work, I think it's important to remember to stop and spend a few minutes talking about the words that we use as scientists, because often the words that we use with each other have a meaning that they don't have when the public uses the same words. Synthetic is one of those really loaded words that triggers responses in people. So in order to get these modules that we can move into our SynBio chassis systems in order to express them in cells, with details, we can more or less look to all of nature thanks to super cheap DNA sequencing. There's a huge natural parameter space that we can look to in order to get these components or these regulatory modules that we want to use in our SynBio systems. So, I mean, that's everything from the bacteria in our body to bacteria that we've been using for 10,000 years to make yogurt, to bacteria with important functions like photosynthesis. And of course, the whole top of this kingdom of life that I'm showing you there, it's all single-celled organisms. All of multicellular eukaryotic life is, it's down here in the corner, which is really, really fascinating. And again, for those of us who works on microbes, no surprise. But for folks who don't work on microbes, this is a pretty cool reminder about how much of life on Earth you can't actually see with your naked eye. And all of this is fair game for building massive databases of natural DNA sequences that can be used for synthetic biology. But wait, there's more. Of course, today we are in the AI revolution. And if your family is anything like mine, they think the only use for AI is ChatGPT and pirate poetry, which is true. But in my opinion, the killer app for AI has been in biology. So you might remember in July 2022, DeepMind, which is a Google company, released AlphaFold, which solves a protein or predicts a protein's structure from its sequence. They released a database with now contains the predictions for the structures of nearly all proteins known to humans. That's more than 200 million of them. If you're old like me, you might remember what solving protein crystal structures has been like for many, many decades. This is obviously a big, big leap forward. In July 2023, using a different model, there were researchers who combined AI with their protein biology in order to design and test new biomolecular and cellular structures with new functions. So that's really exciting. In May 2024, DeepMind released AlphaFold 3, which, given an input list of molecules, AlphaFold 3 generates their joint 3D structure predictions, revealing how they all fit together. And it also models large biomolecules like protein, DNA, and RNA, as well as small molecule ligands. So that helps us, of course, when we're talking about drugs and drug categories. What interested me was that AlphaFold3 can also model chemical modifications to these molecules. So again, really interesting stuff for health and disease and food and more. And now a newer, even newer algorithm from DeepMind can generate novel proteins that bind to other proteins. So again, huge component, a huge potential here for us moving from being able to look at the parameter space that nature has spent 4 billion years on this planet exploring to now what we think are entirely new parameter spaces that have not been explored by life on Earth. And of course, this is why last year, this team, along with David Baker from the University of Washington, won the Nobel Prize in chemistry for this. I get so excited about this research. I think this is the killer application for AI. So thanks to AI, we can also start to access entirely new biomolecular and cellular structures with new functions that, to our knowledge, had never been explored by nature on Earth. So again, David Baker and his group and his collaborators did a lot of work with this, not using transformer algorithms like the DeepMind folks did, or sorry, that I misspoke, not using the same type of algorithm that the DeepMind folks did to do AlphaFold. But because biology is a nested series of languages in a really important way, a lot of folks think that the next frontier for AI in biology is large language models, right? And again, so David Baker and others have done some work in this space as well. So, you know, the number of proteins that could theoretically exist is probably somewhere around 10 to the power of 1300. Not all of these possible amino acid combinations will result in biologically viable proteins, but it's probable that evolution has only explored a small corner of this parameter space here on Earth. In terms of the actual biology, there's lots of genetic engineering approaches that can or will need to be applied in order to unlock the full potential of synthetic biology, including synthetic biology for space. Some of these techniques are agnostic to the target that you're trying to make, and others depend heavily on what you're making and how it interacts with the native functioning of your cell. So I think that the design, build, test, learn cycle is going to continue to be super important to constructing efficient cell factories that have widespread applications. And so this is things like using computational tools and databases in order to guide precise pathway engineering approaches. We'll need to be doing some more fine tuning of expression elements like promoters and RBSs, as well as protein degradation tags. I expect to see some pretty basic protein engineering to change or to improve the properties of the key enzymes in a pathway that we want. I think we're also going to need to control metabolic flux more accurately and more efficiently in our microbial systems. So methods such as synthetic scaffold building, modular engineering, and CRISPR systems, and genetic circuits have also been developed. Now, CRISPR systems have wide applications, not only in genome reconstruction, but also in metabolic network reprogramming, which I think is going to be crucial for looking under the hood. And then, of course, machine learning and other forms of artificial intelligence will enable us to achieve predictive engineering and hopefully to optimize our microbial cell factories for use on Earth and also elsewhere. So hopefully by now I've either convinced you or reminded you that synthetic biology is awesome, not just for Mars, but for Earth as well. So what you should be asking here is, so what's the holdup? Why am I out advocating for synthetic biology for space? And the answer, as with so many things involving biology, is it's complicated. So being able to carry out biomanufacturing depends strongly on input availability. And for space in particular, we're talking about or upcycling of mission products as well as in situ resources. So each space mission class will have a unique set of inventory items. So habitat assemblies and furnishings, functional hardware and appliances, scientific equipment and tools. And many of these inventory items will either be sent to Mars ahead of time or they'll be transported alongside with the crew. In addition, each mission will have different components that are available through the local environment. So the local environment context, the environmental context, really strongly informs the specification of the feedstocks and the processing pipelines that we'll have for in situ synbio-based manufacturing, right? So what it means is that the details of these missions are really, really important. And we don't have the details yet. So Niels, whose picture I'm showing you here, Niels Avarish from our SynBio working group, has really spent a lot of time digging into this. He considered several mission design scenarios and calculated what's known as the equivalent systems mass for each. So equivalent systems mass allows mass, volume, power, and crew time to be converted into a single metric, which is in kilograms equivalents, to predict the up mass requirement as a proxy for expense. So remember I told you at the beginning that it's really, really expensive to take with us everything that we need. If you're going to budget for this, you need to know how expensive this is going to be. So equivalent systems mass has become a standard metric for comparing biomanufacturing systems, but it can't account for aspects such as risk, sustainability, recyclability, complexity, modularity, reliability, robustness, resilience, readiness, scalability, which is always the tough thing with SynBio, as well as safety. So the bottom line here is that the details matter a lot. The mission details matter a lot when we're talking about using synthetic biology in space. This is a complex problem, but it's a solvable problem with a focus on mission architecture. The problem is, if you're a synthetic biologist, you're trying to set up a system based on a context that you can't actually see because it hasn't been established yet, because there's still no real go-to plan that says, we will be going to Mars in the year 2033. This is the vehicle we're going to use. This is what's going to have on it. And so biologists, this is what you have to work with. It's really tough to innovate that way. So, however, we believe that microorganisms can play pivotal roles in the development of technologies for sustainable human exploration of deep space. Again, I'm just focusing on the exploration phase here. Over generations, there's a bigger picture about using SynBio for space for long-term human habitation, if not permanent. So even though I believe microbes can play pivotal roles for these things, we need mature biotechnologies and bioprocesses that enable us to use near closed loop operations of mission functions. So things like life support. And I think we also need to reduce supply chain dependencies for the expansion of a human presence in space. Supply chains in space are incredibly long, incredibly fragile, and incredibly expensive. We need to reduce our dependencies on those supply chains, and biology is a great tool to do that. So microbes need controlled environments in order to achieve the functions that we desire for them. So if we think about food and medicine in particular, we can sort of see what that looks like. So here, I'm showing you something that Dr. Lynn Rothschild is working on here, which is an astropharmacy. It's a single set of biological tools and equipment that can be used to manufacture small doses from a huge library of customized protein and peptide-based drugs on demand, as needed, nimbly and quickly. So if you stock your mission with hardy and genetically engineered spores of, say, Bacillus subtilis ahead of launch, you could freeze dry the spores like seeds. You could save them for years without your refrigeration. And we know that Bacillus spores are space resistant. So of course, when needed, the crew wakes up the spores in a bioreactor, the cells secrete whatever they've been programmed to make, like peptide drugs, and then you purify them and use them. So early on, you'd probably be making the synbiospores themselves on Earth. And then you would take with you a library of thousands of spores, all of which are programmed to produce different drugs that the astronauts are likely to need. So then you would end up with these long haul space missions that are essentially fully stocked with these biological based medical supplies. But of course, long term, what we really want to be able to do is to do the genetic engineering, the synbio itself off Earth. So you could have, again, a library of thousands and thousands of these engineered into very lightweight radiation resistant bacterial spores. So our working group identified 3 important breakthroughs for Mars synthetic biology that we think will also greatly benefit the bioeconomy here on Earth. Number 1 is bioreagents that are shelf stable, that don't need a fridge or a freezer. For Mars, this is important because you'll have less upmass, less complexity, and it won't be as important to have a reliable energy source. And this would also be a great boon for low resource settings on Earth to take advantage of biomanufacturing and synthetic biology. The second thing that we think we need is specific hardware for on-demand biomanufacturing, including small batch on-demand biomanufacturing in space. So for space, this means we would have better flexibility and resilience. And for Earth, this would enable things like personalized medicine and effectively producing materials in small batches. So we think this hardware must be able to maintain control over temperature, pressure, liquid and gas compositions, obviously. We need to be able to collect data about the engineered system itself, as well as the microbes that are in it. It'd be great to have on-demand sample collection. Autonomous operation, or at least partial autonomous operation, would be great. And of course, when we're talking about off-Earth environments, the containment level needs to be rigorous enough for planetary protection. Very, very important. And the third thing that we think we need to enable space and Mars synthetic biology are really robust microorganisms that can serve as a chassis. These ideally are microorganisms that can survive extreme conditions, the cold, dry, radiation, etc., so that we can enable loop closure. Our group also looked at policy recommendations for pursuing these breakthroughs, including what we think is important in terms of investment for early stage research in these things. We believe that both the public and the private sectors are crucial to set the stage for a sustainable and prosperous space bioeconomy that uses synbio as an enabling technology. I'm going to just look at this very briefly in the interest of time. I want to go a little bit more into the chassis organism bit because I think that's really interesting for this audience. So there are various considerations when choosing which microbes you want to use as your chassis organism for space synthetic biology. So of course, biological systems rely on initial sources of carbon and nitrogen and oxygen as inputs to generate biomass. And although there are some organic molecules in the Martian regolith, gas in the atmosphere, which is carbon dioxide and nitrogen, that gas is the most accessible source of carbon and nitrogen. So we also need to consider what the desired end product is when we're thinking about our microbial chassis organisms for space. So biology is quite good at fixing carbons. Autotrophs, right, can make their own food. So we're looking at phototrophs who are using light as their primary energy source for metabolism and photosynthesis to fix carbon. Now, interestingly, of course, on Mars, the natural sunlight is weaker on Mars than it is on Earth because Mars is farther away from the sun. So again, we would need to bioengineer photosynthetic organisms to be able to ideally deal with those lower light levels. So that's looking at phototrophs and then looking at chemotrophs. Again, we're looking for those electron donors for oxidation. So we can do this by harnessing the reducing power of electricity, hydrogen formate, or methane. It's complicated. This paper that I'm citing here has more information about this. So basically, as we're looking at the various types of elements that we need to have fix. We are looking for organisms that are genetically tractable, that preferably have a high metabolic rate for efficient target production, and are robust and resilient so that you, the space synthetic biologist, are spending your time manufacturing and not all of your time troubleshooting and fixing. So it's unlikely that a single bacterium or a single yeast is going to be able to cover all of these bases. So the details are really important. And that's where Dr. Erica Benedictus, sorry, I'm going to go through this. Dr. Erica Benedictus, who has recently founded a nonprofit organization here in the San Francisco Bay Area called Pioneer Labs. And their goal is to engineer the hardy critters, her words, for biomanufacturing that is ubiquitous, reliable, and green on Earth and beyond. So it's really fascinating what they're doing. So they're looking to build a platform, a SynBio platform that enables scientists to mix and match the Mars pertinent characteristics that have already evolved in particular microbes here on Earth. So for example, this is Cladosporium sphaerospermum. It's a fungus that was isolated at Chernobyl. It's tolerant of high radiation doses. It might actually absorb energy from radiation. That's pretty cool. We've got Chlamydomonas nivalis. That's an algae that grows on snow, glaciers, and sea ice. It's freeze-thaw tolerant, and it can grow at extremely low temperatures. This is our old favorite, Deinococcus radiodurans. It's a durable bacteria that grows in the presence of perchlorate, which is a toxin that is similar to bleach, and it's found in the Martian regolith. Today, Deinococcus is employed to help treat nuclear waste. And Xanthoria elegans is a very slow-growing lichen that is successful in polar environments, and it has survived 16 days exposed to the vacuum of space. So while we would expect these microbes to perhaps be a bit more robust to off-Earth conditions, that would make it much easier to grow them in a bioreactor. And you might also see that these microbes and organisms could also have potential for terraforming, were we to decide that that's a good idea for the future of Mars. So again, looking forward, what we really need is research and development to advance the technology readiness levels of the underlying tech for space SynBio. And I'm showing you that here with these pink arrows. I also believe that underlying all of this, we need advanced digital infrastructure for better modeling and simulation of our microbe chassis and what they're actually doing. So open research questions are things like, under what conditions is SynBio most appropriate for enhancing the sustainability of long-term deep space exploration and potentially even settlements? We got to keep planetary protection in mind. If there is extant life on Mars, we don't want to mess it up with our Earth life. I agree with that. We, of course, and then nitty gritty stuff like what technologies do we need to monitor process efficiency? Which of our chassis microbes are the best ones, and how can we cultivate them for a given SynBio process in the relevant conditions of gravitation and radiation and light and all of these things? And then what are the bioreactor and infrastructure requirements that we would need to implement these processes? And then again, how do you maintain these systems? How do you keep monitoring these systems so that you have real-time insight into what's happening? So we have a bit of time. This is the timeline from 2021 for what human space exploration would look like. It's of course already out of date because the Artemis missions have gotten pushed, and we should probably expect to see more of that. But there's lots and lots of information, crucial information, that these missions, and of course the robotic missions as well, are going to give us about things like biosignatures, local conditions, what feedstocks are available, is there water, how much water, and sample return when we do finally achieve that from Mars is going to be really important for shaping the design of these in situ resource utilization processes, because we'll just have a better sense of what's actually there. So lots of exciting stuff coming down the pipeline. I hope everyone will watch that. And so our paper that we published last year takes a deeper dive into looking at space synthetic biology. So I invite you to take a look at it. And of course, the dream here is to look at the very large potential opportunity for innovation in synthetic biology that enables us to use living things as manufacturing centers, as computers, as sensors, as energy sources, in order to give us the food, fuel, and fiber that we need, and hopefully sustainably, for Mars, the Moon, space stations, and various places on Earth. We cannot just transport the way we've done business on Earth to Mars. It's just not going to work. So much of our economy and our manufacturing economy in particular is based on petrochemicals. There are no petrochemicals on Mars. That's it. So what we have, using space as a lens, is an opportunity to really reimagine how we make the things that we need in order to not just survive, but to thrive. I'm really, really excited about this. I hope you are too. This is how you can stay in touch with me after this webinar. Remind me that we met here at this webinar if you reach out to me on LinkedIn. There are a lot of random people on LinkedIn. I don't want to talk to them. I want to talk to you. So please remind me that this is how we met. I've also got a new Substack where I look at the ways biotech is affecting life, and I invite you to come join us there. And the last thing that I want to invite you to do is to consider joining us at the Humans to Moon and Mars Summit, which is in Washington, D.C. at the end of May. If you just Google Explore Mars, you'll find us. I'll be there. I'm running an innovation panel. There's lots of really great energy in this community, and I think that biologists have a really important place in that community, and I look forward to more of us being really present and vocal in the space community. I'm going to stop there. Thank you so much for joining us today. Marshall, do we have any questions that folks want to talk about?
Certainly, we have time for a few questions, but I wanted to say what a great presentation. It's not only a fascinating topic, but it's also surreal in the sense of what is coming, not only in the future, but what we are working on presently. So thank you once again. A lot of people are saying thank you in the chat bar as well. First question right off the bat, what excites you most about the potential of synthetic biology in supporting a sustained human presence on Mars?
Gosh, I only get to pick one thing, huh? Okay, so I mean, you probably heard the excitement in my voice when I was talking about using AI in synthetic biology. I am so excited about the potential for entirely new biological structures and functions that, to our knowledge, have never happened here on Earth. And I actually think that there's a window of opportunity for using that in space in a way that might be harder to sell to the public here on Earth. Not because of the technology, but because of the way people feel about their bodies, the way people feel about living things. And I think if you're talking about a planet that, as far as we know, does not have a biological ecosystem that can be destroyed, I think people just care less about that with Mars than they would care about the unintended consequences or things like that on Mars or on Earth. This is a hypothesis for me. Now, of course, as a biologist, I hope deep in my soul that there is life on Mars, right? That's why I love Mars so much as a biologist. Like that's where the action is for me. Maybe not now, but maybe sometime in the past. And so for me, I would still really want to see us protecting Mars from Earth just as much as we protect Earth from Mars. But I think there's a real potential with Mars as the target to experiment more aggressively with what's possible with biology, simply because we're not carrying our human cultural and emotional baggage with us. I would love to see in the chat what folks think about that. But yeah, I think that's my top answer.
And yeah, and that's the psychological aspect of it is huge as well. And not was a great answer. I think you summed it up perfectly. Um, so this is, you mentioned potential, and there's a lot of unknowns and future items associated with this field. What is one piece of advice that you would give students interested in this field or studying this field currently?
Oh, so I think the piece of advice I would give them is to remember that a way of thinking is far more important than mastering the nitty-gritties of particular techniques. Now, why do I say that? I say that because the underlying technologies that we're using are evolving so quickly that putting all of your energy into getting really, really good at using one algorithm, we have no idea what's going to be possible computationally in 5 years. We don't even know what's possible in 6 months. Just last week, I was giving a talk in Dubai and I was sitting in the back of the room and I was using ChatGPT to write something. And I was shocked by how much better ChatGPT was from 2 weeks earlier. That's how fast this technology is moving. So I would encourage students to work on their mindsets, to remember that they're going to be lifelong learners, to remember that the tools that we're handed today are not the tools of tomorrow. And I would also encourage students to remember that it is very likely that they will have jobs that have not been invented yet, right? Space synthetic biologist, yeah, that's a job now, not for a whole lot of people, but for a job. But an in-space synthetic biologist, that doesn't exist yet. What does that even look like? I seriously doubt someone on the surface on Mars is going to be pipetting by hand. There's going to be something else there. I don't know what it is. So for students, this is a really exciting opportunity for you to be building the future that you're going to be stepping into because it doesn't exist yet.
That's that's excellent. And yeah, it's a repetitive topic of the future is so unknown, but you need to take action in the present to make sure there is the future. Um,
Yes, and just to speak to that for a second, I also encourage people not to think about a single future. Instead, think about a range of possible futures. Some of those possible futures are more positive, and some of those possible futures are more negative. The steps that we take today help bend our trajectory towards preferred futures and away from less desirable futures. So, don't feel like you've got to do one thing today and know what's going to happen in 20 or 30 years. That's unlikely. But you can do stuff today that makes it more likely that the thing that you want will actually happen.
exactly. And for those with sort of unanswered questions, we're sorry that we have a lack of time, but if you want to, you can not only email Dr. Vora at her email address there, or you can email at info@microbe.com, and that way we can get you the answers for those questions that you might have associated with this webinar and this topic. And of course, we would like to thank you once again for joining us, and thank you, Dr. Vora, for presenting such a fascinating topic.
Thank you so much. It's such a delight for me to talk to this community. You probably saw I went deeper into some of the biology than I usually get the chance to do. So thank you so much for giving me the opportunity. And please come play with us in the space community. We want you.
Of course, and that was great. Thank you once again, and I hope you have a great rest of your day.
Thank you. Have a lovely day, everyone.